For my experiment, the use of autofluorescence to image Neotoma livers, we specifically focused on Neotoma because we know that this species can regularly consume a diet full of naturally occurring toxins without causing apparent harm to themselves, which is something that we look at in my overall project. But for this experiment specifically, we're trying to determine if autofluorescence can be used in place of histology to image wood rat liver cells. If we're able to use autofluorescence to image these liver cells, then in the future, we would be able to compare images of liver cells in a control and creosote diet. From there, we will be able to determine if consumption of natural toxins does or does not affect these liver cells. To determine the use of autofluorescence, we used 4 different methods. The first being that we put the liver samples through an ethanol and PBS series, and then stained the samples using a Hoax and ESNY protocol, and imaged the samples using a confocal microscope. For the next 3 methods, we changed it up a little bit, where instead of putting the samples through an ethanol PBS series and then directly staining them, we actually put them in glutaraldehyde for various amounts of times to kind of see if the glutaraldehyde helps with the autofluorescence. After that, we stained them with one of the stains, such as the Hoax and ESNY, the Nano Red, or the Viability dye. If not stained, we then imaged them on the confocal microscope directly. The results are shown below, but before I go into the results of the samples, I actually wanted to show you the diets that the wood rats consumed. For the wood rats that were on a controlled diet, the controlled diet consisted of rabbit chow, which is shown in 1A. And for the samples that came from the treated group, they actually consumed a diet of creosote, shown in 1B. The images to the right are those that are visible through the naked eye, and the images to the left are actually those taken using a confocal microscope. As you can tell, there's lots of detail and lots of contrast. You can see colors such as red, blue, and green. For the samples that were fixed in just glutaraldehyde and did not have any staining done to them, we were trying to see if the glutaraldehyde helped with the overall fluorescence and the clarity of the samples. As you can see, the sample in 3A was left in glutaraldehyde for only 1 night, and the image is still dark, still blurry, and doesn't show a lot of fluorescence. Whereas in comparison to the sample in 3B that was left for 1 night longer in the glutaraldehyde, it showed a bit more clarity. The nuclei is a little bit more defined, and there's slightly more autofluorescence. For the future, we hope to continue trying to see if this trend continues the longer you leave the samples in glutaraldehyde, but so far we've seen that the longer the sample is in glutaraldehyde, the better the image comes out. For the samples stained with Hoax and ESNY, we did have some problems, but with Hoax and ESNY staining, as you can see in 2C and 2D, we actually had the samples be overstained and oversaturated, which gave us the problem that we weren't able to distinguish between the nuclei and the cytoplasm. That's kind of a problem. We won't be able to use this stain method in the future, because what we're trying to see is the nuclei so that we'll be able to count it and measure it. If we're not able to do that, then this staining method does not really provide much use for us. Although we did notice that for the samples that did not become overstained, the amount of time that they spent in glutaraldehyde did help with the fluorescence and the clarity of the image. Although once again, like I said, the overstaining does make us kind of not want to use this stain. One of the problems that we did find in our first sample is we found a parasite. This caused the sample to be a bit more brittle and hard to image. This also could have been something that caused the sample to not be quite as fluorescent and quite as bright. You can see the parasite right here as the little arrow is pointing. It's this little green fellow right here. Now for the next stain, we actually used a viability stain. This stain is one of the ones that proved to be more useful, as it differentiated between the nuclei and the cytoplasm through color coding. As you can see, the nuclei is stained red and the cytoplasm is stained green. This will be very helpful in the future, as it'll help us to quickly be able to count how many nuclei we have and how many healthy versus how many abnormal nuclei we actually do have. The last stain that we used is a Nile red stain, which was another stain that's very useful and one that we will be using in the future as it provided lots of contrast. We were able to readily see the nuclei and the cytoplasm. This is very useful in the future as it'll help us when we're measuring how big the nuclei are when comparing the nuclei from a control and creosote diet. The only drawback about this stain is that when there is a high lipid content, it tends to overwhelm the nuclei, so you're not able to actually see the nuclei as seen in 5A. You are able to see the lipid as it fluoresces yellow, but that's about it. Everything else isn't very visible. So for samples that have a high lipid content, we won't actually be able to use this stain. Overall, though, we were able to determine that the best images resulted from livers fixed in glutaraldehyde for 10 days and that were stained with the viability or the Nile red stain. So using the images from these two stains and glutaraldehyde, we will next be able to compare cells from animals eating a control diet to those eating a toxin-rich diet. We will do this by using ImageJ, which is a program that allows us to measure and count the number of abnormal cells versus the number of healthy cells. What this number will tell us is whether or not the consumption of natural toxins influences wood rat livers. All right. Thank you for coming to my presentation.